Welcome again. Thanks for joining me, and our usual Wednesday afternoon, we'll talk a little bit and then I'll try to answer questions. The first thing is if somebody could put up the link to the video that Stefan Lenkas came out with. I'm actually not sure who produced this, whether it was Stefan himself or Dean, the person who's been moderating our little panel, health freedom or something talks that we've done. The video, and I would say I apologize that it's in a computer generated voice, which is because I think it's because the person speaking, real speaking, was in German, and so they basically translated it with a computer or something. Anyways, that's a little bit annoying, but it's sort of a compilation of the things that I've been saying and some of the slides that Andy has made and Stefan's work, his new experiment. Can somebody let me know something happened here, if you can still hear me? It looks okay now. So there's the link that we just put up, and it's something that's meant to be something you can share with people who are not familiar with, how do we find a virus and show that it causes disease? And there's the whole description with slides and references to Stefan's study, which, as he says, refutes the basic underlying premises of virology. So that was an exciting development. The other thing this week, on Tuesday, on the Subscribe Star thing, I actually had people watch a 8 to 10 minute video of an interview with a, quote, viral immunologist named Byram Bridle. He's from, I think, Guelph, Ontario. And if somebody could put, I didn't let you know that, but the link to that interview in these notes here. I don't know how many of you have actually watched that, but I heard it went, quote, viral. Funny thing to say. When people ask me why I got rid of my cell phone, I just say, well, it got a virus and I didn't want to get sick. Anyways, so it was very interesting. And obviously this guy, Byram, he was saying he was doing this interview, and he was probably seemed a bit shaken by the whole thing because the group that he was with or is with, international collaboration of a number of scientists who are looking into issues around the spike protein in particular. He was claiming that he has new evidence that is really rocking his boat, that they were able to find the spike protein in the blood and different organs, particularly the blood vessels and the brain and the ovaries and the uterus and the testicles and the adrenal glands. And this was shocking to him, and he wanted to let the world know that there's something very wrong with these, quote, vaccines because of this. So I thought I would just speak about this for a minute, because if you listen to it, you also may be surprised that I'm asking you or suggesting that people listen to it, something where the first words out of his mouth is that I'm pro-vaccine and all that. So that's not why I'm having you listen to it. And there's a lot of things about this about his interview that I don't agree with. And there was a lot of things in that he said that literally makes you shake, or made me shake my head and think, you know, we are on different planets here. I don't know if the audio is still good. I think so. I just got a message. So for instance, he talks about that what we're talking about is the spike protein. Now, I'm sure everybody knows by now, the spike protein is essentially the docking mechanism. Well, we're talking theory here. That this is the conventional view. The spike protein is the protrusion off the coronavirus that you've all seen, SARS-CoV-2. All coronaviruses, I believe, are supposedly have this spike, and the spike protein allows it to dock, essentially like a spaceship docks into a whatever command module, I think. And you know, it's frankly a bit like this sort of sexual act. The spike protein is the male part, and it docks into the receptor, and that allows the virus to inject its business into the cell. It's actually very much, as I've said, like a rape scene. So the spike protein is necessary for they say, SARS-CoV-2 to do the damage. And so they found that it was because it was necessary, they could use it as the antigen, meaning they could somehow use it as the part of the virus which they could inject into you and make antibodies. And if you had antibodies against the spike protein, you would prevent this docking from happening. That would prevent the virus from injecting its genetic material, i.e. its business, into your cells, and then you wouldn't get sick, and therefore you would be immune. That's the conventional theory. And he was saying, now we have evidence that this, so that's the conventional story. So what his finding was, and is that they're injecting the spike protein either in a viral vector, that's Johnson & Johnson, or in a lipid nanoparticle, that's Moderna and Pfizer. Or actually, I made a mistake there. They're not injecting the spike protein, they're injecting the mRNA sequence that supposedly codes for the spike protein. So the trick here was to stabilize the mRNA sequence, because typically mRNA sequences break down in nanoseconds. You inject that into a person, you use all kind of sophisticated technology like PEG and lipid nanoparticles and hollowed out viruses, whatever that is. And then you inject these synthetically made carriers with this sequence of mRNA, so it stabilizes, then it makes the spike protein, and then you make antibodies against the spike protein, and then you're good. What his, quote, dramatic revelation was three. First, to his incredulous as he was about this, he said, we now have evidence that when we inject the vaccine into somebody's arm, it doesn't just stay in their arm. I tell you, when I heard that, I thought that was what planet is this guy on? Because we've known, I don't know, 50, 60 years, you inject aluminum and it ends up in your brain. There is no possible way that anything injected in anybody's arm only stays in their arm. How or why or on what basis he thought that was true is just unbelievably naive. Anyways, so to his shock and surprise, he's finding evidence with his group that this mRNA sequence gets out of your arm and somehow goes to other parts of your body, like, and starts making spike proteins, apparently, in your blood vessels, in your ovaries, in your testicles, uterus, adrenal gland, nerves, crosses the imaginary blood-brain barrier, etc. So this is horrifying to him, because then the next thing he discovered, I don't think he discovered it, but he was reporting on, is, lo and behold, stop the presses. It turns out the spike protein is itself a toxin. And he said, quote, nobody would have predicted this, he said. Nobody thought for a moment that the spike protein could possibly be a toxin. Well, there's been a number of us who've been saying this for probably over a year since we've got into this, but apparently that's not the people he listens to. And so he cannot possibly imagine that the spike protein could be a toxin, but he's finding out there's evidence that there is elevated levels of spike protein in the blood. I'm not sure if this is actually true, because I know Andy and I have not been able to find confirmation of that, but he says there's new data that shows that's true, which I wouldn't be surprised. So the injection of the mRNA makes you make spike proteins. The spike protein is directly toxic to the endothelial lining of your blood vessels, where it has these so-called ACE2 receptors. So it binds with that. It binds to the platelets, causes blood clotting, sometimes bleeding, other times either too many clumping of the platelets or not enough function of the platelets. That would cause bleeding. And it causes an inflammatory, destructive response in your ovaries, leading to infertility and ovarian failure. And that's the reason why it binds to receptors that are also in your uterus, causing miscarriages and menstrual bleeding and all the other symptoms that we're seeing, and also in your brain and other nerves, causing neuropathy and other neurological damage. And according to old Byram, nobody could have seen this coming. Nobody thought the spike protein could possibly be a toxin, which is unbelievably naive and ridiculous. But there you go. Now, the other thing that we've heard a lot about this week is, we've heard a lot about it for a whole year, but somehow more this week, was the engineered virus. Where did this virus come from? And we're now closing in on dear Dr. Fauci, who may or may not have had something to do with what is called gain of function, which means essentially making the virus more virulent. And so we're about to catch them, and even the mainstream media has got onto this, and we're about to nail these guys red-handed. And then you see it all over the anti-vax world. See, we were right. We caught them red-handed, gain of function, all that stuff. Now, as you know, I think I have a different take on this. So here's my best hypothesis as to how this whole thing came about. So for probably decades, 20, 30, I don't know how many years, they, and when I say they, I mean the people who are sort of perpetrating this fraud, decided they wanted to find a bioweapon that would have a large and varied array of toxicity in the human being, on the blood vessels, for fertility, menstrual function, neurological function, etc. And so they started trying to engineer this bioweapon in all these labs all over the world. Now, it's not so difficult to find a protein toxin. There's a lot of those made from bacteria, made from snakes, venomous insects, there's all kinds of protein toxins. The trouble is how to stabilize that and deliver it, because proteins need the watery environment that they're found in to have the right shape so that they have the right activity. And then if you try to give the code, the sequence of mRNA that would make the protein, that literally lasts nanoseconds. And so you inject it, 1 second later it's gone, doesn't do anything, you get no protein toxin made, and the whole thing didn't work. Same with injecting DNA. This is a technological nightmare, and the reason it is because it doesn't work like that. mRNA is not a stable molecule, and so you can't just inject it because biologically it doesn't work. So they had to spend probably decades creating a effective bioweapon, i.e. protein, and B, more importantly, the delivery system. What do you put it in? What kind of nanoparticle? How do you actually make these nanoparticles? If you say you're going to put it in a viral protein, how do you do that? You can't get a guy with a scooper who goes every day and looks at this virus and takes a little watermelon pick and scoops out the genetic material, then a syringe, and injects it in. You know, it's nothing like real world stuff like that. So they spent probably decades on developing the toxin and the delivery system. That's what they've been doing in these labs all this time. My guess is, while they haven't got it perfect, they figured they had something that they could roll out, and the toxin was probably the spike protein. So they have a synthetic mRNA, which they could stabilize, and that could allow, or force is a better word, your body to make many, many copies of this toxin that would then have all these effects on your blood vessels and nerves, ovaries, testicles, uterus, adrenal glands, etc. So that was basically the plan and the technology to carry that out. And then you would say, obviously, because apparently you can assay for spike proteins in the blood, the question then is, what do you use for a cover story? Because you can't just make people sick and they say, why did I get sick? And then somebody eventually figures out that they have too many spike proteins in their blood or in their blood vessels or uterus. And then they would ask the obvious question, where did this spike protein come from? I don't know. This must have got it. You must have ate a bunch of spike proteins. Well, nobody thinks that. So enter the virus story. So how does the spike protein get into you? Well, it gets into you because the spike protein is attached to the imaginary virus. And so it's actually, it's not our fault. You just got infected with a virus that has a spike protein, and we had to try to deal with this. So we injected you with something to make the spike protein perfectly innocently, because we wanted to make you make the spike protein, which we thought was just a docking mechanism and had no toxicity. Whoops, turns out the spike protein is the toxin, and this engineered mRNA to make you make more of it is the cover story. The virus is the cover story. The engineered virus is the patsy. And I don't think anybody, I don't think most of the people who are speaking about this actually understand what happened. And one of the reasons I think that is over this past year, and believe me, I've had lots of conversations with prominent voices who are speaking out against the vaccine and out against some of the lockdown measures and speaking out against some of the things which I appreciate and applaud. But when I ask them, why do you think there's an engineered virus? Or what is the proof that there's an engineered virus? Can you show me an engineered virus or any virus from even one sick person? Believe it or not, their answer is, well, how do you explain gain of function research? Now, I can tell you, if the answer to how do you know that a virus exists is because people are doing bioweapon gain of function research in laboratories, then you are seriously confused about the whole field of virology. And that gets into my next point. There's been a lot of people attempting to debunk us and say Tom is nuts and wrong, and we're sure now that this is an engineered virus unleashed on the world, and we're sure about pathogenic priming. And I know this probably gets confusing for most of you, or many of you, or maybe some of you. And the only way to deal with this is to go back to the basics. How do you know there's a virus, and how do you know it causes disease? And you will eventually get them to admit that this so-called engineered virus has never been found in any single person with COVID as of now. No published evidence that this is true, not one. And what you hear when people are asked, and I've seen this, so have you heard this, the people saying there's no virus at all? Well, yeah, but that's not true. Then they start launching into the genetic stuff. Well, we don't do it like that anymore. We sequence the genome and then we pull out different sequences and we match them to the database and then we put them together. And the next thing you know, nobody knows what the person is talking about. It's even questionable whether they know what they're talking about. And everybody's confused. And all I can say is when that happens, just keep asking yourself, but where did they get these sequences from that they're so supposedly finding? So the answer is they found them either from an unpurified specimen from a human or almost always actually from the results of a tissue culture. And then ask yourself, and maybe ask them, if you get a sample of RNA from a human lung, how do you have any idea where that sample came from? On what basis are you saying that that could have only come from a SARS-CoV-2 virus? On what basis do you know that it didn't come from human lung cells, which have, for instance, the same sequences that we call the sequence for the spike protein? How do you know it doesn't come from the other millions and billions of microbes, including supposedly viruses, that live in our lungs? How do you know it doesn't come from the fungus? In the cell culture, how do you know it doesn't come from the fetal calf serum? How do you know that these sequences that you're talking about don't come from the breakdown of the monkey kidney cells? After all, we have clear evidence now that the breakdown happens whether you add a virus or not, and that no virologist can distinguish between that which arises from the breakdown of a monkey kidney cell or a human lung cell versus an exogenous virus. It simply is not possible. So you have to be very rigorous of keeping to the process, and don't get confused by all the genetic stuff and the sequencing stuff and the technological, speak and technobiology, which has nothing to do with why humans get sick or what is happening with them. So that's a bit of a rant. And with that, I can get to some of the questions. First question. Please talk about what may be in the mass and test swabs, and can they be harmful to us? This is something that Andy has brought up to me, and he actually sent some of the mass and the swabs to see if they actually have spike proteins in them or the sequences. As far as I know, we don't know that. I wouldn't be surprised, but I would hate to say that I know that there is something in there which I don't actually know at this point. What is the role of the furin cleavage site in the SARS-CoV-2 spike protein? Is this site proof that the protein has been engineered? I don't know that I could say that. The protein was engineered. That cannot possibly have come from any virus. Thank you for making strephanthocid available on our website. Do you think it has utility in helping stop the blood clotting and heart inflammation occurring from taking the jab? It's an interesting question. Strephanthus and the active ingredient wabein have no effect that I know of on blood clotting, which is why it can actually be used with any kind of blood thinner. Like aspirin or Coumadin or Plavix or any of the other ones, not sure if Xarelto. So there's no contraindication that way. As far as I know, nobody has suggested or shown that it has any effect on bleeding time or clotting or even platelet aggregation, although it does make the platelets and red blood cells more flexible and able to move better. Would it have effect on the inflammation? That may be, because it improves the metabolism of the heart, and I would expect that it might have an effect on reducing inflammation, but I don't know that for sure. In these independent injection analysis, there hasn't been many of those, and we've been trying to get them, and I don't know of any very clear independent labs that have come out with here is the data. But have you heard of them finding something like just saline or nothing? I've heard of that, but I don't have any proof that that's the case. I've heard that there was 2 that we did. The injections were just saline. And I wouldn't be surprised, because it would be a great way to reduce the amount of side effects. So if Aunt Bessie says, yeah, nothing happened to me, it could be that Aunt Bessie got lucky and got saline. Recently, someone told me that he tested positive for COVID-19, but suffered only a couple days. He was told that his children who were asymptomatic passed it on to him. My question, is it possible for a healthy person to make another one sick? Answer is no. This is a complete fantasy. There's no science to this at all. There's no science even to what it means to test positive, because if I assume it was an RT-PCR test, which is not a test, it's a manufacturing step. And since nobody has ever shown that the primer sequence actually come from a virus, there is no way to tell that this test has anything to do with any virus. How do scientists support their claim that spike proteins on virus are the mechanism by which viruses invade our cells? That's an interesting question. I'm going to take a guess here, and maybe I shouldn't be doing that, but it's kind of how I live most of my life. At least I tell you when I'm taking a guess. So here's the part of this that's provable, i.e. observable, is a better way to say that. they don't see any of the viruses in a normal person or in a sick person. They don't see them in the beginning of a cell culture, and then they inoculate it with unpurified stuff from a person who's sick, and then they see lots of these particles. So that's what you can observe. No particles in a sick person or a normal person or in a normal cell culture. Then something happens, and then you see into the cell culture, and then you see lots of particles, and those are called the viruses. Now, you have to make up a story of how that happened. In other words, how you got from nothing to see to lots of these particles. And the last story you want to say is, well, you just blew up the tissue, and so it makes inevitably every dead and dying tissue makes lots of these particles. You don't want to say that, because then you'd say, oh, well, they're no big deal. We don't have to worry about it. So you have to make up a story of how the virus attaches to your cell, injects its DNA or RNA business into the cell, takes over the cellular machinery, causes the cell to do its bidding and replicate itself hundreds, thousands, millions of times, and then it emerges victorious after its conquest, buds out of the cell, taking a little bit of the cell as a souvenir with it, and then you see lots of it. So that's the viral theory story. Now, none of those steps have actually been seen, and when I say seen, I mean videoed, like in a movie. So what you may see, first of all, there has to be a docking mechanism, or else how would it attach? So since you see, or since you postulate that there's this protein sticking off the end, that must be the docking mechanism. And then you can probably find, X-ray crystallographic evidence that if you have this spike protein and that it attaches to an ACE2, quote, receptor, which is actually just another protein, you can find that mixture, and then you postulate or hypothesize that the ACE2 receptor must be, or ACE2 protein must be, a receptor embedded in the cell membrane, even though you can't see it and couldn't find it. So, but it must be there, because else this wouldn't work. So, and what you do see is complexes from, X-ray imaging, X-ray crystallography imaging, and probably electron microscope cryo freezing stuff. You probably see complexes of ACE2 proteins mixed with spike proteins. And so that becomes the only observable fact, and the rest of it you make up. And I think that's how they got there. Okay, are you familiar with natural non-invasive remedies for a blocked tear duct? I had some clown doctor operate on me and it closed right back up. It depends on the age. I remember for very young children, what we used to do is we would put our finger there and we would roll up and squeeze out the mucus that was solidifying in the tear duct, and you would feel a little popping, and that would clear out the duct, and then we put chamomile compresses on it to keep it warm and keep it flowing and keep it from being inflamed. And it happened with my daughter, and that's how we fixed it. Whether that would happen with a grown adult, I don't know, but that's still what I would try. I would use compression, and you just go from the nose and roll up towards the eye. And then you would put hot compresses with chamomile tea, and that's the only thing that I've ever seen that worked. Can you explain what an autoimmune disease is, and are they all expressions of the same thing? Also, are you still writing a book on this topic? So I used to believe in autoimmune diseases until I read the work of Harold Hillman, who doesn't believe in autoimmune disease. And it would be difficult for me to recreate his argument right now, but I think the essence of it is that antibodies are not, first of all, they're not specific for any pathogenic virus, obviously, because pathogenic viruses don't exist. And so why would you make antibodies to them? So my current understanding of antibodies is, as I've said, it's the fifth step in the detoxification pathway. In other words, you get poisoned and then you poop it out. That's step one. You get poisoned, you try to poop it out, but some of it gets into your bloodstream. So then you pass it through the urine and then you pee it out. You pass it through the kidneys and then you pee it out. That's the first backup to the liver and the bowels. First liver, then bowels, second kidney and urine. And if you still have things to detoxify, then you sweat it out through the skin. So that's backup plan number two. If that isn't enough, and now you have toxins essentially destroying your tissue like your tonsils, then you recruit bacteria to the area to essentially biodigest the dead and dying tissue. And we erroneously call that a bacterial infection. So that's essentially backup plan number three. When that doesn't work, and now you have your bloodstream is full of toxins or has toxins that you can't eliminate, then you make antibodies, which are proteins, to bind with and neutralize these toxins so that they can be handled by the kidneys or the liver. So we erroneously think that the antibodies are attacking your tissue, but what's really happening is they're just a reflection of how poisoned you are and how toxic you are, especially with proteins. And so we have erroneously thought they were the agents of disease, but in reality, it's the toxin which is the agent of disease, and the antibody is just your body's way of neutralizing it. And the more antibodies means the more toxins. And if you would rather not have to get antibodies involved at all, you would rather not have to have bacterial infections, you would rather not have to sweat every day, that's not so bad. The first three are sort of normal. And I wouldn't say you'd rather not have to pee, because that's normal too. You'd rather just stay with the first three, and that's called being healthy. Once you get into 4 and 5, you're talking about now you have a problem, and you either have so-called bacterial infection, which is just the bacteria eating the dead and dying tissue, or it's even deeper, and now you make antibodies because you're poisoned. Which gets into the next question, my thoughts on tetanus. It seems that many who know the dangers of vaccines are still unsure about tetanus. And yes, that's true. And in fact, I was unsure about tetanus for a long time until I read the work of, I don't know if she's Russian or Ukrainian kind of immunologist. I think she is Tetiana, and I'm not sure how to say or even close to her last name. And she did a wonderful piece on tetanus. And it really enlightened me as to what the real story is. So here is what she found, basically. So again, it's always helpful to go through the regular theory first. So the regular theory is, here you are normal, and then you get a wound, usually a puncture wound, and that's important because a wound that has no air, so it's an oxygen poor environment. And then you have a bacteria that always lives in spore form in the soil, and I believe it's called Clostridia tetani. So that spore of that bacteria gets inoculated into the anaerobic without air environment in your foot or wherever, and then that is the perfect growth medium for the bacteria, and it grows, and then it makes this toxin called the tetanus toxin, and the tetanus toxin leaks out from the wound into your bloodstream, binds to imaginary receptors, although they don't think they're imaginary, and causes you to have muscle, tetani. It causes essentially a permanent contraction of your muscles, so they contract, but they can't relax. And so you have this jaw clenching or respiratory failure because the diaphragm, which is a muscle, can't move. And that's because of the binding from the tetanus toxin to the receptors on the lungs or the jaw or any muscles of your body. And that's how it kills you. Now, what's interesting about what she found is that while it is possible to clinically reproduce the symptoms of tetanus in laboratory animals by the injection of pure tetanus toxin into an animal, that has been done, at least as far as I know. I would just also point out that throughout all this, I've said, and we've said, that what the problem is with, quote, infections is not the bacteria, but some bacteria themselves make toxins, just like snakes do and humans apparently do, lots of them. And the toxins can be toxic, meaning poisonous, and can even kill you. There's botulism toxin, tetanus toxin. So this doesn't prove the germ theory. It just proves that there's many different ways that you can get toxins. You know, food poisoning is the ingestion of staph toxin. So there's nothing unusual or germ-like. It's just that this particular toxin came from the germ. Okay, so what's the problem with it? So the proof is that you can inject pure toxin and cause somebody to get, or at least an animal to get tetanus. At least I think that's true. So, but here's the problem. When this has been attempted to experimentally reproduce tetanus in, I believe, animals, maybe people, they inject tetanus spores into a wound and nothing happens. And so they put tetanus spores and they mix ground shards of glass into the wound. And interestingly, still nothing happens. And then they inject the tetanus spores, shards of glass, and a poison called carbolic acid, which degenerates the tissue. And then the people get, or the animals get tetanus. So it apparently is not so simple as just being made, at least in nature, because in normal clinical situations, there is no pure tetanus toxin. That never happens. And so apparently in an animal, it's not so easy to give somebody tetanus. You have to actually put glass shards in their wound and actually put some sort of poison in there. The other thing that then became true is, and I checked this myself, at least used to be there, if you go to the CDC website and look up tetanus, they have a very interesting comment, which is clinically, you cannot rely on the presence of tetanus toxin to confirm or deny that the person has clinical tetanus. Nor, they say, do antibodies assure you that the person doesn't have clinical tetanus. So what does that mean? So the reason for giving somebody a tetanus shot, which is essentially the tetanus toxin, is then you make antibodies to that toxin, which I wouldn't be surprised if you do. And the antibodies then confer immunity, because anytime you're exposed to that toxin again, you already have the antibodies or you have the blueprint for making them. You make the antibodies, bind the toxin, and it doesn't hurt you. But interestingly, the CDC says that if you have somebody you think has tetanus and you want to know whether they do or not, you cannot rely on the presence of toxin in the blood or in the tissue to help you answer that question. Meaning, some of the people with tetanus have the tetanus toxin, and some of the people with tetanus have no evidence of the toxin, which, if you think about that, is bizarre. What do you mean? So how did they get tetanus if they have no evidence of the actual agent that causes the illness? Now, interestingly, I would admit that that's not proof that the toxin doesn't cause tetanus, because it could be that the toxin is degraded again within nanoseconds, like most proteins are. And the reason you can't see it with a blood test is because it's been degraded. So while that's an interesting finding that you cannot find the toxin in at least some, if not many people with tetanus, that doesn't prove that it's not there or wasn't there. But the other one is very interesting, which is you cannot take somebody who you think might have tetanus, right? So you have somebody who's got their jaw paralyzed, that's where lockjaw came from, or can't breathe, and so you have to put them on a ventilator because they have paralysis from exposure to tetanus toxin. And if you do a blood test and say, oh, well, they don't have antibodies, therefore it's more likely they have tetanus, or conversely, they have lots of antibodies, so that means they for sure don't have tetanus. They warn you that you can't use that as evidence either way. In other words, somebody could or couldn't have tetanus and have no antibodies, and they could or couldn't have tetanus even if they have a lot of antibodies. It doesn't help. And then I looked up, where did that come from? What evidence do they have that even people who have tetanus antibodies, which is, after all, is the whole point of doing the tetanus shot, that these antibodies aren't protective? And I found a very interesting paper. I don't have the reference here, but you could find it just like I did, where they, back in the day before recombinant tetanus antibodies. So now if you've never had a tetanus shot and you go to the emergency room after a wound that you might have tetanus, and they're worried that you might have tetanus, they give you not a tetanus shot to make you make antibodies. They just give you the antibodies. Now, back when I was in an ER doctor, we used to do that by giving horse serum antibodies. And we were told that the incidence of anaphylaxis and death was somewhere between 5 and 10%. In other words, if you had 10 people in a day who you had to give this thing called hypertet, which I probably had 5 my whole career, if you had 10, you were going to kill one of these people fairly immediately just because of the injection. So believe me, you do that with a good dose of trepidation. But so that was in the days when they used horse serum. Now they make the tetanus antibody with yeast and recombinant DNA, so they don't need the horse. So it's not so dangerous. It's still dangerous, but not that. But in the days even before the horse serum, they used to do this by hyper-vaccinating human volunteers. Hyper-vaccinating means they gave them over and over again tetanus shots to make them make more and more antibodies. And so then they would get enough antibodies to harvest, and they would purify them, and they would use that as the treatment for somebody who needed antibodies and didn't have time to make them. And the amazing article I saw was they had this guy, I think it was a male, who he was hyper-vaccinated and had thousands of times the elevated level of tetanus antibodies. And then anybody want to guess what happened to him? I would love to see the answers here. Maybe I'll look at the chat for a second. He got tetanus. Exactly. He got tetanus. So you guys are learning. Meaning the antibodies didn't work. So that was a long story, but it is a fascinating tale. My conclusion is vaccinating people to make them make antibodies has not been shown to work. And there's something else going on here. I'm not exactly sure what it is, but antibodies not the answer. Ready for enlightenment. I have a question about the 30-minute heart cooking that cardiologist Manel Ballester talked about yesterday. This is a podcast interview that I did with the Spanish cardiologist Manel Ballester, who is the main person who's carrying on the work of a guy named Torrent Guasp. I think that's right. And this is an incredibly fascinating study of how the heart is actually formed, and out of understanding the form, you can then begin to see how the heart functions, and then you can get an idea of how the human being is organized and functions. And everybody has to watch this interview. I will admit that some of it you won't understand, just like I didn't, even though I've been dealing with this hard stuff for 30 years. And then Manel and I spent another hour afterwards that we didn't record, just trying to figure out how this all works. And I still can't really get it, but someday, hopefully I will, and I'll be able to explain it. But this is absolutely revolutionary stuff. So was the heart disconnected from the body before boiling? Yes, the person was dead and they took the heart out and they boiled it for 30 minutes. Was it a cadaver? I mean, it was a dead person, and they did it. He had an arrangement with the Spanish hospital that when people died, they would call him and he would come and get their hearts as soon as possible so they could work on as fresh hearts as possible. Can a trained surgeon unroll a live human heart to repair it? I think the answer to that would be no. Next question. If there exists a successful way to package and deliver this spike protein through items such as nasal swabs or masks, then wouldn't anybody who wanted to get this protein into us be able to put it in any product en masse, such as Q-tips, etc.? So there's also a difference between the vaccines don't have the spike protein in them. As far as I know, none of them do. They have this sequence, the mRNA sequence, that makes you make the spike protein, even though that's not really the way it works, but it's sort of the way it works, and it's the way they're trying to make it work. So again, the problem is the delivery mechanism. And I don't know that you could deliver it on a Q-tip or in the water, because the mRNA would not be very stable. The spike protein would not be very stable, and I don't know that you could do that. What do I think about C0M from pine needles? I have nothing against pine needles. I think it's whether anybody knows that it helps stop inappropriate mRNA sequencing, I think that's a huge stretch, but I have nothing against people drinking pine needle tea. We have a lot of pine trees on our land, and I started drinking a little bit myself just because I thought it was an interesting idea, and I like it. Whether it makes RNA sequencing appropriate, I have my doubts, but maybe. Any idea why metals are sticking to the head in the unvaccinated? I don't know what you mean by that, so I'm going to skip that. If there is no protein spike issue, I didn't say there was no spike. The spike protein is a toxin, maybe the toxin. No shedding. Their shedding could be of the delivery device, the nanoparticles or the vectors, if they could get them to be stabilized. I'm not sure if they have. The only way you would find that out is take vaccinated, measure their levels of spike proteins before and after the vaccine, and then do the same with people who were not injected, measure their spike protein levels in their blood, which apparently you can do, or maybe in other tissues, and then have them be exposed to somebody and see if they make more over the days. If they do, then something was transferred. I would love to see that study done, but as far as I know, it hasn't been. I have a friend who was against all this, but her husband got the shot right away. Her teen daughter had menstrual changes. Then my friend started getting menstrual changes, plus having pins and needles, and it's been over a month. That was before anybody was talking about this. That's another anecdotal story, which these are valuable inputs, and this should be investigated, as I've said over and over. How can there be a spike protein if there's no virus? Easy. You just make the mRNA sequence, attach it to a yeast or a E. coli, and turn out millions and billions of spike proteins. How can something that exists for nanoseconds be preserved? You put it in a lipid envelope so that the body can't get at it. They must have made it in a laboratory. They did make that in a laboratory. Gain of function must be making the toxic proteins. Absolutely. That's what they're making, not viruses. Why are the COVID cases decreasing? Probably because they turned down the PCR amplification cycles, which gives them a very clear way of saying, now we have less cases, which is why it's one of the favorite tools of all tyrants. So last question, same line. Is it possible the spike protein was created directly to put into the vaccine? They're not putting the spike protein, they're putting the mRNA sequence. The spike protein was the goal, to make you make spike proteins. Everything else was either the cover story or the delivery device. So they now have a delivery device, apparently successful. I had my doubts, but that would make you make more and more of this spike protein, and that's a real problem. So somebody says, bring your own swab to a test. There's even a better answer, which is don't do a test. They're totally useless and probably harmful, and there's no information you can get on a test, so don't do it. Okay, everybody, we will see you next week. Sorry again, I didn't get to all your questions, but thanks for joining me and have a good week. Thank you. Bye-bye.